So, extracellular matrix. So again, where is all this water being stored? Oh, one thing I forgot to mention when I was talking about the glycogen, someone asked me a question that jogged this memory, was that a big kind of thing to instill in people if you have patients, when I get third years, fourth years coming back from rotation, they're like, I can't believe how much of what I'm dealing with is basically poor diet, right? And so one of the things to help people kind of stay healthy if they're trying to do a diet, because the big issue is that they keep dropping off it, is that, when you don't eat, you're losing water weight and then they rebound up. That's because of glycogen. So for every pound of glycogen you have, it holds 3 pounds of water. So when you don't eat, you start using that glycogen. It has all those OHs, so there's all this water bound. So when you use that water leaves you. And that's why you pee. And so then the first time, let's say you do a 1-day fast or even morning time. Let's say you do a 1 or 2 day fast. You'll lose a ton of weight, mostly water. As soon as you eat, you build up glycogen and you store a lot of water. So if you're fasting in the first beginning part, you probably won't be dehydrated because you're actually releasing a lot of water. And when you're coming off a fast, when you eat, you're going to suck up a lot of water, so then you would want to have more water. Okay? All right. So I will point out how many high-yield disease states are here, okay? So we're talking about the inheritance pattern, symptoms, explain the biochemistry of Marfan syndrome, Williams, MPSs, mucopolysaccharidosis, scurvy, osteogenesis imperfecta, Ehlers-Danlos syndrome, emphysema, Graves' disease, Menkes, Alport. Also, Hunter and Hurler syndrome and Gaucher disease, Tay-Sachs, and I-cell. So a bunch of diseases we're going to cover in the next 2 hours. So if you spend a little more time on this lecture, probably not wasted time, high-yield content, okay? And don't get crazy and say you want this all the time, but I made some of these objectives kind of study aids in that I just gave you the big take-home answer. What's happening in Marfan syndrome? Your fibrillin's down, okay? So I imagine you might do that with objectives as you're going, so for some reason I did it for those. Okay, so a quick recap, because we haven't gone over the organelles yet. I gave you a quick cheat sheet. This is actually, I think, from the Khan Academy. And the one that I want to focus on here is the lysosome on the bottom. So that is your recycling center, okay? And so not today, but later on, I'll talk to you about fasting and increasing autophagy. Autophagy is kind of your cell in a controlled way recycling things, and so it's using the lysosome there. And so we're going to talk about a lot of things today where the lysosome's not working right? So you have, basically we're a bag of water, then we have all the structural stuff that lets that work, so that the structure, if you think of that like I-beams or mesh, there has to be a lot of that to hold everything in place in this bag of water. Well, all of that mesh and I-beams has to somehow be maintained and metabolized. That's through the lysosome, okay? And so when you have these enzymes that don't work there for whatever reason we'll get into, that's all those diseases I mentioned, or that's a lot of them, okay? So the first one I'll talk about quickly is Graves' disease. So it is different than the other ones, okay? So it is different because, and the only reason I'm including it here is because the end result of Graves' disease is an increase in these things called GAGs I'm going to talk about, Glycoaminoglycans. And that's basically these large carbohydrate structures that can hold water, okay, in the extracellular matrix. And so Graves' disease causes an increase in them. It's an autoimmune disease, which increase dermal fibroblasts, which end up increasing GAG production, okay? I think I have more information there, okay? That's the take home though. So then we're gonna talk about mucopolysaccharidosis, MPSs, okay, and there's a few of them. Those have caused non-functional lysosomal proteins. So you might have a genetic defect that causes the protein, a lysosomal protein, to not be expressed, to not work, and that causes an increase in GAGs because you're not metabolizing them, right? You're not breaking them down. There's Sphingolipidosis. This is a class of diseases. We're not going to really cover much of this today. I'll cover Gaucher. But that also causes non-functional lysosomal proteins, okay? So this is lipid defect type. Then there's oligosaccharidosis, and then there's mucolipidoses. So these three in the middle all have non-functional lysosomal proteins. The mucolipidoses have proteins that are unmodified, and so they don't get sent to the lysosome. So they could work, but they're just not being put into the right spot, okay? And so all of these ones are a decrease in lysosomal function. Okay, so we're going to walk through these three types of extracellular matrix. There's the basal lamina, the ground substance, and fibrous protein. And so here are my emoticons to remember what each of them are. Okay, so the basal lamina, so here's your epithelial layer, okay? And so the outside, like that could be skin, right? Then right below that, you have the basal lamina. Then most of this picture is this orange kind of background, that's the ground substance. And then the darker lines in it are the fibrous protein, okay? And so the basal lamina I have here as a TheraBand, and you should think of it like that. It's a thin supportive structure, okay? And so here's a picture in green of it wrapping nerve cells, okay? It can be underneath skin. It can be wherever there's kind of a layer of support needed, okay? And it's made up of laminin, which is each one of these colors in this left structure is a single polypeptide, and so it forms a triple helix, okay? And it has various domains, but the important stuff is it can interact with itself. So it can form a network, okay? And so that's how it creates a sheet of support. Does that make sense? Okay. So now we're going to move on to the basement membranes and GAGs, glycoaminoglycans. So they have this repeating structure of an acidic sugar and an amino sugar times some large N, right? So here you see I boxed, again, you do not have to know the structures. We're going to talk about these words, so they're different, and those will have different diseases. So you see the red thing I boxed is the acidic sugar, okay? And then there's a gray box that's an amino sugar. And that can also be sulfated, okay? And so you have hyaluronin, dermatan sulfate, chondroitin sulfate, keratin sulfate, and heparin sulfate. I'm boxing some differences again. Hyaluronin does not have a sulfur group, and keratin sulfate does not have an acidic sugar, okay? The other one thing I'd point out is that hyaluronin, unlike the other ones, is not synthesized in the Golgi. It's actually synthesized in the cell membrane. So you can think about, this is going to be a long thing, right, with a lot of N. You can imagine that kind of just being excreted out from the cell membrane. Okay, so here are what GAGs look like. So they have a core protein, this purple protein, but the rest of this picture down here is all carbohydrate, okay? So I talked to you about hyaluronin being made from the membrane. That's this green line, okay? So the core part of a GAG here is a long hyaluronin, okay? Then there's a little adapter to this core purple protein, and then off that come hundreds of chondroitin sulfates. And so you don't really need to know it, but there's a serine off the core protein, a linker region, and then that repeating disaccharide. The point is, these little blue things, there's a ton of them. And what are those? Those are a bunch of OHs, okay? So they are going to swell with water, okay? So this is how we are bags of water. This is the outside of our cells. What is there? Mostly this. You can also have membrane-bound ones. So here are proteoglycans, the three you need to know, syndecan, glypican, and perlecan. The difference is in their anchoring. Perlecans are actually secreted, so they're not bound to the membrane. They can actually, we'll talk about things like integrin, they can interact with the extracellular matrix, okay? Syndicans have anchor right through the membrane, so they're locked in, and then glycans have a lipid anchor. So we'll talk later about modifications, or actually I think I already hit that. Did I already hit GPI anchors? Yes, no? No? Okay. So you can have a GPI anchor, so you're basically attaching a lipid to a protein, and that lipid is going to insert in the membrane. That's another way of anchoring the membrane without the protein having a helical structure or something to go through. Okay, so they all have heparin sulfate, and then syndican also has chondroitin sulfate. Okay, turn and talk. I didn't tell you this, but I want you to think out what you think the answer is. Okay. 10 seconds. I'll admit, this one was a very tough one, I'm not going to lie. Oh, guess not. Okay. So let me go over this take-home, then explain why I thought it was tough and how you got it right. So the take-home here, remember, is that when something, let's say a protein is an enzyme and it's acting alone. If that's the case, if you have half of it, you're still fine, right? So that would be recessive. It's not dominant. You lose one, you're still fine. If things are acting together, like a laminin would be an example, it's forming a network. If half of it's messed up, but binding the other half, then you're messing up the whole thing, so then that's dominant, right? This one I thought could throw you off because it's not... Some of these structures, they're large structures, so it looks like it's happening all together, right? But what is actually going on is often you are having an enzyme that might degrade them, and you're losing one of those enzymes. So you still have the other enzyme, right? So that's why it's recessive, and you guys obviously knew that. Okay. So we're going to go through how many? 6, I think. 6 of these. Again, you do not need to know the structures. So chondroitin 4 and 6 sulfates, these are the most abundant GAGs. Let's see if I can, whoops. Okay, so they're the most abundant GAGs. They're found in tendons, ligaments. They're found in the aorta, so you can have heart problems. Actually, wait, I think I visually I'm showing you some of this. Yeah, so you guys see all this, right? It's not hidden. They have non-covalent interactions with hyaluronic acid, and they bind cartilage and can make a tight network, okay? Dermatin sulfate is kind of, you should think of as more flexible. It's found in the skin, blood vessels, and heart valves. And you can write down whatever you want. I think I gave you backups here. Yeah, so I gave you my non-horrible notes as well. Okay. Okay, so keratin sulfate has 3 types. Mainly 1 and 2 are the most important. So, yep. So keratin sulfate 1 is found in the cornea. 2 is found in loose connective tissue. Loose connective tissue. And then 3 is found in the brain. So this is the most diverse GAG. So some of these are going to have the phenotype or how the patient presents may be a wide range of things, okay? Hyaluronic acid it's found in bacteria. It's also found, it is very, it swells with water a lot, so you should think of that as like a lubricant. So anywhere where there's impact, it can be there. So shock absorbers in the synovial fluid of joints. And yeah, heparin sulfate. It is found intracellularly in mast cells, so in white blood cells. It's also found in the lining of the liver, lungs, and skin. And heparin sulfate is found in the basement membrane. I think we already talked about that. And I think I have, let me clear this. This might be one of the things I did this morning, but I think I have that, written for you guys. Okay. All right. So let's slow down and talk about GAG degradation. If you are wanting, so the notes have just continued to grow for this lecture, so use them with caution. But also a great resource for this lecture is Lippincott's. Have you guys used Lippincott's yet? Biochemistry? No? Show of hands, who has looked at Lippincott's Biochemistry? Not many. Okay, that's the main book I would recommend for my lectures. If you feel like way over encumbered, then you can look at The Cell. That's kind of like undergrad basic biochem, and then Lippincott's is much more clinically oriented. So if you understand Lippincott's, I'd go there. So anyway, there's a bunch of figures from Lippincott's in here. Okay, so I tried, any questions before I move on? Okay, speed, fast, slow, okay. Tell me if I'm too fast. You want me to go slower? Slower, okay. All right, so I tried to summarize this. I would read through this slide, but I tried to summarize it for you easily. So basically, you have this large carbohydrate molecule, and you're starting to metabolize the GAG, right? You're trying to break it down. And so here I show what you're doing. In this step, you're taking off a sulfur. In this step, you're taking off a sugar. Taking off a sulfur, taking off an acyl chain, taking off a sugar, taking off a sugar, taking off a sulfur, okay? At each point, if you don't have that enzyme, you have a disease, a genetic disease, okay? So the first one you don't have is Hunter syndrome, right? You're not taking off the sulfur. The second one is Hurler syndrome. Then you get to this disease, Sanfilippo syndrome, which was originally classified as the same based on presentation, but now we know it's basically 4 different diseases that all manifest similarly because they're in this chain. And unfortunately, the letters they were given are not in order. So it's A, C, B, something else, and then D. But you don't really need to know that as much. But the point is they are 4 different diseases that are classified together. And then Sly syndrome's the last one, okay? So now, they're not super, initially I had this on a ton of different slides, but it just kind of seemed to go too long, so I tried to put it all on one. I'm gonna fade in color underlines that are the same here. So this is, for patient presentation, what is the same with all these, okay? So, developmental disabilities, all 4 of these boxes, okay? They're going to be to varying extents, and I made you a sweet review table, if I do say so myself. Okay. Then there's this little line, corneal clouding. Okay, so Hurler syndrome has that, Sly syndrome has that. I put a green line here because Hunter syndrome does not have corneal clouding. Okay, and they have MPS numbers too, right? Hunter syndrome MPS 2, Hurler's MPS 1. Okay. So what can take enzyme replacement? Hunter syndrome and Hurler syndrome are shown here. And then I have a whole bunch in red. I would point out that Hunter syndrome is X-linked deficiency. unlike the others. Okay. In the bottom left here, do you see that viral therapy is ongoing? And there's 2 links in the US and in the EU. So I've given continuing education talks to physicians, and they've come up afterwards and not known how to find a clinical trial for their patient, which kills me. Okay, so if you ever want a clinical trial in the US, it's clinicaltrials.gov. Okay. In addition, if like this link, it has a URL to clinicaltrials.gov with a search term, I think for glycoaminoglycans, I forget what it is. So that is always live. It always goes and researches it. So this will tell you the current clinical trials for these diseases, and the same is for the EU. They both are kind of active live. While I'm on that topic, clinical trials, because sometimes I'll talk about policy to you guys because you guys are going to be the ones to help shape policy. If you go forward, if you're approved in clinical trials, you now have to report the results. That's great. There's kind of a standardized way we can see that on clinicaltrials.gov. It's still the case that if you pull out of clinical trials, like there's side effects or something happens, you'll find a ton of clinical trials that don't have data, which is just killer to me because there's all this known information that the greater community cannot learn from. And so that is, I'm guessing lobbyists from pharma basically thought of that as proprietary knowledge that they didn't, want to share. But it's very frustrating when I actually have my college roommate this year, his son was diagnosed with Sanfilippo syndrome, which is extremely rare. So, there's clinical trials that you don't have the information of what went wrong. So we'll get into all that. What is the likelihood of these? Hurler is probably the most frequent, so at 1 in roughly 100,000. Hunter, again, X-linked is 1 in 160,000. These numbers vary a little bit based on the study. Sanfilippo looks like it's more prevalent, but it's actually a group of 4 diseases, remember? So combined, they're 1 in 70,000. And then Sly syndrome, it was hard to even get that number because it's so rare that people have a problem predicting the frequency because different countries seem to vary so much because it's just very rare. Okay. So those are recessive because those are enzymes that are deficient. Any question about those? Yes.
So for the Sanfilippo, you said combined there's 70,000? 1 in 70,000?
Yes, combined. And I think I made a mistake. Oh, no, I didn't. In that later on, I'll have a table that has this information, and I broke up the percentages by each one. Yeah. Sorry, the question was, actually it was recorded. Sorry, just yell it out. I'll repeat it.
If you give us, during an exam, what the problem is, or if it's Sanfilippo syndrome, should we know what that chain looks like? Or should we just worry about,
Do you mean the digestion biochemistry pathway?
Yeah, exactly.
Yeah, so I'll speak to this when I show the grid. You should know how the patient presents, what enzyme is deficient, what builds up because of that, and then what is the treatment, okay? Later on, I don't know if we're gonna have time to get to this, but in the new, I was trying to give you guys more quiz questions and clinical presentations. In the very end, I have some cases, and you see this little emblem up here? I would think of clinical cases, I do anyway, I would think of them in this sense. Can you define it? What are the risk factors? What are the symptoms and signs? Diagnose it, or clinical differential, and then treatment. That's basically being a doctor. Okay, so if you know those, then you know the disease. And so I kind of sometimes say, like, this case is about risk factors. Yeah.
So do you get decreased severity of symptoms as you move down the chain because it's not enzyme effect at that level?
No. Yeah, that's not how that works. Let me show you that chain again. So some of that has to do with where the aggregation's happening. So Sanfilippo, for example, is tough because a lot of that's happening in the brain. And so there's huge hits there. And in addition, when we talk about viral therapy, some of these are harder to tackle than others. So Sanfilippo is probably the hardest because, one, viral therapy hasn't worked a ton yet, and also you have to get it into the blood-brain barrier, or past the blood-brain barrier, or inject it directly in, or whatever. Good question. Okay. Okay, so moving on to eye cell disease. So here we have modification of proteins, okay? So from the ER to the Golgi, you're adding sugars, in this example I'm giving you, to N amino acids. Amino acids have a nitrogen, okay? So asparagine. You can add sugars on O-link stuff too, threonine and serine. And then they can get modified and mannose phosphorylated, okay? So here you're adding a phosphate group. If you don't have this enzyme that does that adds the phosphate group, you end up with eye cell disease, okay? If you have phosphorylated mannose, that moves the protein to the lysosome, okay? So this is an important targeting mechanism that you're failing to get because of this last step in the modification, okay? So what happens when that doesn't happen, when you don't phosphorylate it? Well, it's not getting sent to the lysosome, so instead it goes to a default secretion to the extracellularly, so you can detect enzyme in the blood, okay? The lysosome can't break down lipids, sugars, and GAGs because you're not getting enzymes there. You end up with abnormal cell architecture, and you have early developmental problems. So 2 years, you have skeletal abnormalities. You can also have cataracts. Okay. So again, if you think of that little tree structure I showed you, you can see how I'm covering a few things there. You're covering what's the definition of eye cell, what enzyme went wrong, what happens, where does stuff not go, what's the presentation, right? Okay, a quick review of coagulation. So you're going to definitely get this later. Basically, you have factor X activates thrombin, which creates fibrin from fibrinogen, which causes clotting, okay? Antithrombin 3 blocks those. It blocks 2, 9, 10, 11, and 12, but 2 and 10 are the big ones, okay? So antithrombin 3 blocks that. And heparin, which is modified with these sugars, binds antithrombin 3 and increases its blocking activity 1,000-fold, okay? So this is how heparin can block clotting, okay? Because of that modification, the carbohydrate added on. If this sugar is not here, then the protein structure of antithrombin 3 doesn't have the hook to bind. It's binding onto that carbohydrate section. Okay, so some lysosomal storage diseases, Gaucher disease, you're not having glucocerebrosidase, which is breaking down this. So you end up with glucocerebroside, which is a glycolipid, and you get enlarged blood cells, white blood cells, macrophages, okay? Again, you don't need to know that structure. I'm just showing it to you. Tay-Sachs, you don't have acetyl hexosaminidase A, which causes you to increase ganglioside, which is a sphingolipid. I'm just giving you these two. Those are the only two I would ask about now, but I wanted to give you that big picture of you're digesting things down to ceramide in the lysosome, and at each of these breakpoints, you have a different disease. I just talked about the center one, Tay-Sachs and Gaucher. Okay? I think I have more information there. Yep. All right, so when you're thinking about these genetic defects, think about, this whole path only works for a genetic defect, right? So there's some inheritance pattern, you wanna know that. Is it autosomal recessive or dominant? What is the mutation? Because the mutation can make a difference in the therapy you give. Okay, so what happens normally? You have DNA, you transcribe it, you make RNA, that's spliced, that's translated into protein. That protein goes on and does something. It can be inhibited by something, by an inhibitor. If it works, the enzyme takes some reactant to some product. Okay, this is the generic view of what's going on with proteins. And at each of these steps, if it's not working, you can have a therapy. If this is the problem at the DNA level, gene therapy is the thing you want. We're working on a bunch of those. In 15 years, we should have a very different view. A lot should be done, but we're not there. Read-through inhibitors are a kind of really important thing that are not talked about a lot. I'll mention them more later, but in long story short, if you have I think I mentioned this in the protein lecture, if you have a nonsense mutation, if you have a mutation that causes a stop codon too early, let's say you're supposed to be 300 amino acids and it stops at 50, that's going to go off and be degraded. A read-through inhibitor allows the molecular machinery to pass by that like 10% of the time. So it gets past the one mutation, keeps going to the next stop, which is the correct, and then you get enough protein to not have a phenotype. Amazingly, that means you're going past the normal stop codon in all your other genes 10% of the time, but the read-through inhibitors are only used for diseases where your life expectancy is lowered. So any hit of those, I've tried to look up side effects of read-through inhibitors, and they seem to be low enough that there's a benefit whenever people use them. Again, think if you read through the stop codon on a normal one, usually that's way too big, and that's going to end up being degraded anyway. So you're kind of probably just using a little too much ATP. Okay. There are drugs starting to try to aim at splicing, but they're not really doing a lot yet. But that is another place you could go where if you have a genetic defect at one of these points that causes a splice problem, you could potentially correct that and get the right protein. There's small molecules that help chaperone, so they help proteins fold. Cystic fibrosis is the poster child of that. You can have specific mutations that just make your CFTR not quite fold correctly, and if you can add a drug that makes it fold well enough, it gets into the membrane and you're fine. And then you could do substrate reduction. So if the problem is that this enzyme isn't working well enough, you could try to reduce the reactant so that's not building up, right? If this green Pac-Man isn't working well, the problem is often a buildup of this reactant. Now that won't help if the problem is not getting the product, right? And then enzyme replacement therapy, that is dependent on delivery mechanism where the enzyme needs to be, right? If it's a digestive problem, that's great. If it's something in the brain, how are you going to do that? Does that make sense to everyone, generically? Okay. So down here, there's a link, and then there's another link off screen to what newborn screens are covered by state. So this is not federally mandated, so you can type in a disease. So here on the right, I've typed in cystic fibrosis, and basically you see every state covers it except Pennsylvania, and Pennsylvania is a weird one where it kind of does. I forget what orange means, but everyone basically screens for that. Sorry. In comparison, if I look up MPS 1, Mucopolysaccharidosis 1, the red are states where it's not in the prenatal screen. Why does this matter? Because in California, your patient would come in if they had MPS 1 and you wouldn't be diagnosing them based on a phenotype. You would just know that from their electronic medical record, right? Whereas Indiana, surrounded in blue, we do not have that, okay? So in this state, in the clinic, you could have a child that has MPS 1 and you wouldn't know. You'd have to detect that from a developmental delay. And to be fair, these generally move with how actionable is it, right? So if someone has Sanfilippo syndrome, there's clinical trials for some of those now, but there's not much to do. So what's the benefit of putting it in a prenatal screen right now? Right? Okay, so here are some of the tables. These are just review tables I made for you. So here are the MPSs, the common name, the inheritance pattern. So notice the differences, right? So Hunter syndrome is the only one that's not autosomal recessive. It is X-linked recessive. The deficient enzyme, and then accumulated products, okay? Now, this is my crazy table that has a lot of information, okay? But this is basically how I would do a differential diagnosis for MPS. So here are all the different MPSs. And here are the different presentations in columns that they can be, okay? Let me point out a few things here. So MPS 1, there's 3 flavors of that. It can be Hurler syndrome, which is the most severe, Hurler-Shih, which is in middle, and then Shih, which is least severe, okay? And those are kind of defined by phenotype. They could be genetically now because we know what that genome does to the phenotype. And then here are kind of the regions that are affected. So let's start looking. So let me read across slowly once, and then we'll go through. So is there intellect or developmental delays? What's the lifespan average in years? Is there CNS degradation, sleep disorders, corneal clouding? That's a big one that you can detect right on a checkup. Coarse facial features, joint stiffness, recurrent infections, so of the ear, sinus in particular, cardiac abnormalities, hyperactivity, hernias, enlarged liver, skeletal dysplasia, hypertrophy of tissue, hydrocephalus, so enlarged head, and hydrophytalis, so swelling of the fetus. So MPS 1 is very bad, okay? So, and I also have the incidence here, okay? And I didn't get it for all of them, but for most of them I have per 100,000. What's the frequency? Okay, so you have a huge intellectual development delay. And it presents early, okay? So the lifespan average is 5 to 10 for Hurler syndrome compared to Hurler-Shih is 20 to 40, and Shih is 30 to normal. And so basically, again, no color blind people, right? But even if you are, this is all just shading. You basically see it gets worse. It gets not worse and not worse as you go down for those 3, okay? Hunter syndrome, which is MPS 2, you can have slight developmental delays, but not nearly as bad as MPS 1. Again, notice the differences. So there's not corneal clouding, okay? You do have coarse facial features. Then I did all the Sanfilippo's together. Again, notice the differences. So Sanfilippo D or MPS 3D is the worst for hyperactivity. They all have intellectual developmental problems. And again, these are hard to treat because you need to get the viral therapy to the brain. And then Sly syndrome, the lifespan can be normal. It's not as bad as the others. And it's known in the board, you might see hydrophytalis. So the fetus will have swollen. Okay, and I didn't give you these. I don't think I removed it. I'm just listing them for you, but I wouldn't test you on those. All right, there you go. So how can you treat this? Well, if you look here at the distance that someone can walk and enzyme replacement therapy, so they're having problems, MPS 1, 2, 4, and 6, basically you're adding distance as the weeks go on, about 26 to, what is that, 4, 12, 16, 5 to over 5 months, maybe half a year, you're getting an increase by enzyme replacement therapy for these. Here's the cheat sheet I made for you for what can be done for these. So for the MPSs, enzyme replacement therapy is being done for MPS 1, 2, and 4. You'll notice MPS 3 is the outlier because we can't get it into the brain. Can you do stem cell replacement? It is being tried for MPS 1, 2, and 7. Can you do gene therapy? Gene therapy should work for all of them. These are the ones that currently have clinical trials. So even MPS has clinical trials. I don't know where those stand. And then you could, in theory, have synthase inhibitors, right? So to not create the GAGs that you can't degrade, but you know, we're bags of water, you need them. So I don't know how far you could actually go with that. Okay. So now I'm just showing this again to kind of put these in perspective. These are all because you're not being able to degrade these massive things that are swelling with the water, right, causing our synovial fluid. Let me see where I want to stop. I think this might be a good place. 25. No. Okay, let's get through collagen, and I'll go for maybe 5 more minutes, 10 more minutes. Okay. So this you've all had, I hope, in undergrad biochemistry, but I'm going to go through it quickly and then we'll hit clinical cases. Or actually, I'll go through it medium speed. So, so you have collagen, which ends up being this triple helix of a protein. Okay, so every color here is a single polypeptide. When you first make it, this is called procollagen, okay? When you chop off those ends, that's called tropocollagen. And then when you take those clean triple helixes and put them together into a fiber, that's this collagen fibril, okay? Now, one of the things for diagnosis here is that this is a really broad phenotype because there's tons of mutations in the collagen, okay? So there's over 1,000 mutations in 22 genes, and you make up this collagen by combining 2 alpha 1s and 1 alpha 2, okay? That's these colors. There's 2 alpha 1s and 1 alpha 2. So all those combinations mean there's a broad spectrum of presentation, okay? So here is your DNA for collagen. You make your mRNA, you make your collagen, then select prolines and lysines are hydroxylated, okay? That's going to become important. So there's OHs added to them. Then some of those are glycosylated with glucose and galactose, okay? And then you form this pre-alpha chain where you form all these disulfide bonds to kind of keep it together, which I always find weird because you're going to end up chopping them off. But you get that. So that is then exported, okay, with these, and then you get chopped off and you end up with tropocollagen. Okay, so now you have a nice collagen molecule that's formed a fiber. Because of the way that those stack, there ends up being a gap in a kind of repeated pattern. And so in an electron micrograph, you can actually see that structure, okay? That's this gap every boom, boom, boom going down. And so we talk about those in different types according to kind of their structural properties. So type 1 is in the skin, bone, tendon, blood vessels, cornea. Type 2 is cartilage. Type 3 is blood vessels. So those are all fibril forming, okay? So looking like that. Then there's network forming. So that's the 4 is the basement membrane. We talked about what that looked like. Now you know that there's a network forming version there of collagen. And then there's fibril associated, which is 9 and 12, cartilage and tendon. Okay. So this is by far the best pseudonym I've ever come up with. I think first aid I have over here. But for me, I came up with bending to stretch can reap benefits. So type 1 is bone, tendon, skin. Type 2 is cartilage. Type 2 is reticulin. And type 4 is basement membrane. Okay, so there's that again. And so this is a big part of our body. It's 30% of our protein. Okay. Oh, and there's a little video if you want to see how Jell-O is made. It's really corny, but kind of whatever. Okay, so if I was going to tell you, let's go 3 more minutes, then we'll pause, that collagen's this crazy non-globular protein, what 2 amino acids would you guess collagen's rich in? So not alpha helices, not beta sheets. If I were to say, name 2 amino acids that are different, what would you say? Yes, glycine and proline. So glycine makes up 33% of collagen. And if you add proline and hydroxyproline, it starts to get close to that. It's 23%. Glycine has to be in every 3rd position because the triple helix is wound so tight, there's not physically three-dimensional space for a C-alpha carbon. So it only has room for a hydrogen coming off the C-alpha. So every third space, it's glycine, glycine, glycine. Okay, and the sequence tends to be glycine proline something or glycine something hydroxyproline. Okay. So here I told you that proline gets hydroxylated. Okay, that works via prolyl hydroxylase, and you need ascorbate to do that. Does anyone know what ascorbate is? Someone into the mic.
Vitamin C.
Vitamin C. So what happens if you don't have vitamin C? Scurvy. Very good. So I'll just finish scurvy and then we'll take a break. Who has, show of hands, who's seen the show Deadwood? No one? God. They redid, the new one was bad. But anyway, it's a really good show on HBO. Long story short, it's back in like Frontier days, and like whenever they had a big important meeting, he would whip out a can of canned fruit. And it used to be very important because you could get your vitamins like vitamin C in canned fruit, especially when we were seafaring nations crossing the Atlantic, people would get scurvy because they didn't have vitamin C. And so they were having problems hydroxylating their proline, which they were unaware of. So what happens? Your collagen fibrils impaired, you become tired, you have bruises due to capillary frailty, gum disease, you can see, and poor wound healing. It doesn't, yes, that's a nice story. It still can happen. So it can happen in the elderly, largely because they're neglected, so they have poor nutrition. It also has happened in wars. By far the biggest kind of recent one was in the Sudan in 1989, 25% of the population got scurvy due to lack of access from the war. And then more recently in Afghanistan in 2002, 6% had scurvy. Also, I talked to you about kind of populations that were on like rigidified diets to be concerned about. So there's examples from the Church of Scientology where they would only give their children this boiled mixture that didn't have any vegetables. And so they didn't have any vitamin C, and so their children got scurvy. So if you have a patient that has a very restricted diet of any kind, then some red flags should go up of like, okay, what do I need to worry about? Okay, let's pause there, come back at 5 after, and we'll wrap it up.